Before translation, pre-mRNA turns into mRNA by taking out its introns via the spliceosome enzyme and mending together its exons via the RNA ligase enzyme. Note here that mRNA is formed in the 5' to 3' direction because it must be anti-parallel to how the RNA polymerase reads the mRNA strand. Introns are segments of mRNA that are not needed, whereas exons are the portion of the mRNA that codes for the amino acids. After the introns are taken out and the exons are mended, a methyl cap and polypeptide A tail are added on to the pre-mRNA to prepare it for leaving the nucleus via a nuclear pore. We can now see the new mRNA leave the nucleus.
Now the mRNA strand will pass through the small ribosome subunit and large ribosome subunit, starting with the start codon, which is adenine, uracil, and guanine. The transfer RNA will come in delivering a specific amino acid to the ribosomes as the codons are read off the mRNA list. The correct amino acid is transported to the ribosome using the info encoded in the mRNA. tRNA contains a 3-base anticodon that pairs with a complementary codon in the mRNA on the base of the tRNA. The complementary base pairs are adenine and uracil and cytosine and guanine. Once the tRNA reaches the E position, it will exit the system, and the amino acids will bond together, and this will form an amino acid chain, also known as a polypeptide chain. This process will continue, growing the polypeptide chain with each tRNA and amino acid added to the system. This process will stop at the stop codon uracil, adenine, and guanine. This will bring an end to the process and render a complete polypeptide chain. The stop codon ensures that the mRNA message is read in precisely the same way every time.
All cells in a multicellular individual called eukaryotes have essentially the same DNA, yet different cells express different sets of genes, and within a given cell, the pattern of gene expression can change over time. Single-celled organisms, or prokaryotes, such as bacteria, face a more difficult challenge. They are directly exposed to their environment and have no specialized cells to help them deal with the changes in that environment. To meet this challenge, they express different genes at different times. The genetic code is redundant because there are a total of 64 codons but only 20 amino acids, which means that several different codons call for the same amino acid. With that said, it is not ambiguous because each codon specifies only one amino acid. The genetic code refers to the unity of life because it is universal, meaning that nearly every organism on Earth uses the same code, a feature that illustrates the common descent of all organisms. The genetic code explains the diversity of life as there are a countless number of different DNA sequences that can be coded by the amino acids, creating different genes as a result. This is evident through observing our diverse population today and noting the different hair, skin, and eye color of those around us.